Welcome to the deep dive. Have you ever just, paused to think about the sheer complexity, the precision of your own central nervous system?
It's pretty mind-boggling.
Yeah, it lets us do everything from, a simple reflex like pulling your hand away from heat to composing music, or even just finding your way through a crowded street.
Exactly. And what if you could sort of peek inside, understand the brain's internal wiring, the actual building blocks?
That's what we're trying to do here on the deep dive. We take dense stuff, like a chapter from a big physiology textbook, and pull out the key ideas.
Make it make sense.
Right. So today we're diving into chapter 16 of Boron and Boulpaep's Medical Physiology, the focus, those intricate circuits of the central nervous system.
And our goal is really to unpack these complex ideas, make them clear, engaging, and just accessible for anyone studying this, whether you're in college or medical school.
We want to build that understanding from the ground up, connect it to what you might actually see, clinically.
Yeah, how physiology connects to diagnostics, to understanding diseases, even treatments. It's not just abstract concepts.
Definitely not just memorizing facts. It's about getting how the brain works,
Yeah.
from basics up to complex stuff. We'll try to paint some mental pictures, hit those key terms naturally, and hopefully help you feel, more confident.
Let's jump in then.
Okay, first big idea.
Mm-hmm.
Neurons never work solo. They're always in networks, these interconnected circuits.
Right. Even in really simple animals like tiny jellyfish, hydrozoans, their neurons are often multifunctional, doing sensing, rhythm generation, motor output, all in one cell, but they're still connected in rings.
So even simple systems need connection.
It shows the basic advantage, right? Connected neurons are just way more powerful. Then, as animals get more complex, the neurons get super specialized, allowing for incredibly detailed functions.
And it's not just about reacting to the outside world, is it? The brain makes its own noise, so to speak.
That's a crucial point. Neural circuits generate their own intrinsic activity. Think pacemakers, but for neural patterns. We call them central pattern generators, or CPGs.
CPGs, like for walking or breathing.
Exactly. They create these precise rhythmic outputs. Sensory feedback fine-tunes them, sure, but the basic rhythm can start right there in the circuit.
Amazing. Built-in rhythms. So, if the nervous system's job is adaptive behavior, how is it organized? Big picture first.
Okay, highest level, you've got major neural subsystems, things like the visual system, motor systems, large brain areas, highly interconnected, often with information flowing both ways.
And then zooming in.
We find local circuits. These are the neuron arrangements and connections within one specific brain region. They handle the local processing. Think of brain regions as being built from these repeating modular local circuits.
And even finer.
You get down to microcircuits, the really detailed arrangements within local circuits. And finally, individual neurons, synapses, membranes, molecules like neurotransmitters, ions, all the way down to the genes controlling them.
Wow, layers upon layers. But you said most local circuits share some common elements, despite the diversity.
That's right. Generally three main parts. First, input axons. These come from outside the local circuit, bringing information in.
Like sensory info to the spinal cord.
Yep, axons from skin, muscles, joints entering through the dorsal roots, but also signals coming down from the brain or other spinal segments.
Multiple sources.
Always. In the neocortex, you get input from the thalamus sensory, motor info, but the biggest input source is actually other cortical circuits, so key takeaway. Local circuits integrate multiple input types.
Okay, input axons. Element 2.
Output neurons, also called projection or principal neurons. Their axons leave the local circuit to connect elsewhere.
Like motor neurons in the spinal cord going out to muscles.
Exactly. The alpha motor neurons sending axons through the ventral roots. Or in the cortex, big pyramidal neurons in layer V projecting down to the brainstem and spinal cord, while layer 6 neurons project back to the thalamus. Key idea. Multiple outputs, too.
Makes sense. Input, output. And the third element must process things locally.
Precisely. Those are the interneurons, sometimes called intrinsic neurons. Their axons stay within the local circuit. They do the local processing.
And these vary a lot.
Hugely. Some excite, some inhibit, some connect very specifically, others very broadly, and there can be a ton of them.
You mentioned the cerebellum.
Yeah, about 100 billion granule cells, just one type of excitatory interneuron there. It really shows how much processing happens locally.
So it's not always neat categories. Projection cells acting like interneurons sometimes.
Right, some projection neurons have axon branches called collaterals that stay local, and some interneurons don't even have axons. They connect via dendrites. Plus, inputs aren't always synaptic signals. Some neurons sense chemicals like CO2 or physical things like temperature. It's a really dynamic, interactive network.
Okay, got the building blocks. Now let's look at some simpler functions. Those stereotype responses, spinal reflexes.
Right, motor reflexes. Rapid automatic motor responses to specific sensory triggers. Sir Charles Sherrington did foundational work here back in the late 1800s.
He actually coined the term synapse, didn't he?
He did. His work on reflexes gave really strong evidence they existed. Reflexes are fundamental, like building blocks for more complex movements.
Before we hit specific reflexes, maybe a quick refresher on motor units.
Good idea. A motor unit is just one alpha motor neuron and all the muscle fibers it controls. It can be tiny few fibers for eye movements or huge, thousands for leg muscles.
And all the motor neurons controlling one single muscle are called.
The motor neuron pool for that muscle. Okay, so the stretch reflex, also called the myotatic reflex, probably the simplest.
The knee-jerk one.
That's the classic example. You tap the patellar tendon, it briefly stretches the quadriceps muscle.
Oh, leg kicks out.
Right. The response is a rapid contraction of the same muscle that was stretched, opposing the stretch. The circuit starts with sensory info from muscle spindles carried by superfast group IA axons.
And they connect directly.
They make a monosynaptic connection, just one synapse directly onto the alpha motor neurons controlling that same homonymous muscle. Sensory neuron, synapse, motor neuron, boom. Fastest possible circuit.
But there's more to it, isn't there? Something about the opposite muscle.
Yes, reciprocal innervation, crucial part. Branches of those same group I sensory axons also connect to inhibitory interneurons in the spinal cord.
Okay, interneurons.
And these interneurons then inhibit the alpha motor neurons going to the antagonist muscles. So for the knee jerk, while the quadriceps contracts, the hamstrings are told to relax.
Ah, so it clears the way for the main action, makes the reflex stronger.
Exactly, maximizes efficiency. Now, slightly different is the Golgi tendon organ reflex, or inverse myotatic reflex.
This one senses tension, not stretch.
Right. Golgi tendon organs are in the tendons, sensing muscle tension, especially during active contraction, and its usual response is kind of the opposite of the stretch reflex.
It causes relaxation.
Typically, yes. Relaxation of the muscle where tension increased, and maybe contraction of the antagonist. It's about controlling force, protecting the muscle and joint, uses Boulpaep axons and interneurons.
Okay, got it. What about the flexor reflex? That sounds more involved.
It is. Think stepping on something sharp. Ow!
You pull your foot back instantly.
Right. Rapid withdrawal. That involves activating flexor muscles in that leg and inhibiting the extensors. But you also need to stay upright.
So the other leg has to do something.
Yes. Simultaneously, the opposite leg extends activating extensors, inhibiting flexors to provide support. This crossed extension reflex is part of the whole withdrawal response.
Wow, that's coordinated. Across the spinal cord.
Absolutely. It involves a whole network of excitatory and inhibitory interneurons spreading up and down and across the cord. And the stronger the painful stimulus, the stronger and wider the response.
But these reflexes aren't totally independent robots, right? The brain can step in.
Definitely. Descending pathways from the brainstem and cortex constantly modulate reflexes, usually by acting on those spinal interneurons.
Like when you brace yourself to catch something heavy.
Perfect example. Anticipation overrides the basic stretch reflex. Your brain co-contracts muscles to stiffen your arm. Or think about the Gendrasic maneuver in a neuroexam. Clenching your teeth can heighten leg reflexes.
So the brain adapts them, makes them context dependent.
Precisely. Allows suppression, like tolerating a bit of pain, or enhancement when needed. Reflexes are dynamic parts of all movements. And interestingly, the neurons involved in these reflexes are often the same ones used for more complex stuff, like walking.
That alternating flex-extend pattern seems familiar.
It's the basic rhythm for locomotion, which leads us right into central pattern generators, or CPGs.
Okay, CPGs. These are like pre-programmed movement sequences.
Sort of. A motor program is a set of muscle commands the nervous system can fire off without needing constant moment-to-moment sensory feedback to tell it what to do next.
So it can run the basic pattern even as feedback is cut off.
Yes, although feedback is absolutely crucial for making movement smooth, adaptive, and effective in the real world. But the core rhythm can be generated internally.
And these rhythms are everywhere.
Oh, yeah. Walking, running, swimming, breathing, chewing, scratching, shivering. So many fundamental behaviors are rhythmic.
What makes a circuit a CPG? What are its key features?
Well, they generate cyclic, coordinated timing signals They command potentially hundreds of muscles with precise timing. Think flex, then extend, then flex again for walking. They can coordinate multiple rhythms like arms and legs.
Kind of flexible.
Very flexible. They adjust for speed changes, obstacles, different gaits like a cat's walk versus its gallop, and they need reliable ways to turn on and off.
Where are they located? You mentioned breathing CPGs in the brainstem.
Right, but surprisingly, the CPGs for locomotion for walking and running are actually located within the spinal cord itself.
Really? Not the brain.
The core rhythm generator, yes. The classic evidence comes from animal studies, like cats with spinal cord transactions. If supported on a treadmill, their hind limbs can still generate coordinated stepping movements. The spinal cord holds the basic pattern.
Mind blown. Okay, how do they make the rhythm?
Couple of ways. Sometimes individual neurons within the circuit have intrinsic pacemaker properties, like heart cells. They just fire rhythmically on their own.
But that's not the whole story.
Usually not, especially in vertebrates. More often, the rhythm emerges from the synaptic interconnections between neurons. Graham Brown proposed the half-center model way back in 1911.
Half centers. What are those?
Imagine two groups, or half-centers, of interneurons. One group excites the flexor motor neurons, the other excites the extensor motor neurons. Crucially, these two half-centers mutually inhibit each other.
So when one is active, it shuts the other one down.
Exactly. If you add some kind of continuous go signal, or tonic drive, and a mechanism so that inhibition doesn't last forever, like neuron fatigue or adaptation, they'll flip-flop. Flexor's active, then extensor's active, back and forth, rhythmically.
And that can happen without sensory feedback from the muscles.
That's the key insight of the half-center model. The rhythm arises from the circuit connections themselves.
Have we learned more since 1911?
Oh, definitely. We've studied CPGs in detail in simpler systems, like the sea lamprey, that fish that swims with undulating waves.
What did that show?
It confirmed the importance of reciprocal inhibition, but also showed how sensory feedback is integrated. Lampreys have stretch receptors in their spinal cord. Stretching one side excites the CPG on that side.
Helping initiate contraction there.
Yes, and it also triggers inhibition of the CPG on the opposite side, helping end contraction there and start the next phase. Feedback helps smooth the rhythm and transitions.
And coordinating along the body for that swimming wave.
Right. Connections between spinal segments ensure the wave propagates smoothly. For forward swimming, there's a precise lag, about 1% of the cycle time, between segments. They can even reverse the timing for backward swimming.
And the brainstem talks to these spinal CPGs.
Absolutely. The brainstem sends signals to initiate locomotion, control speed, direction, and the spinal CPGs send signals back up, informing the brain about the ongoing rhythm. It's a two-way street.
And these principles, spinal CPGs, sensory feedback integration, brainstem control, apply to us walking too.
Broadly, yes. The details are much more complex in mammals, but the fundamental organization is similar.
Understanding all this circuit stuff must be vital for neurology.
Hugely important, especially for diagnosing where damage might have occurred based on the symptoms. We distinguish between lower and upper motor system injuries.
Lower motor system means the motor neurons themselves.
Yes, damage to the alpha motor neurons in the spinal cord or brainstem, or their axons going out to the muscles. This causes things like localized weakness, paresis or paralysis, loss of reflexes or flexia, decreased muscle tone, atonia or flaccidity, and eventually muscle wasting, atrophy.
And upper motor system, that's higher up.
Right. Damage to pathways descending from the brain in the cortex, brainstem, or spinal cord itself, like from a stroke or spinal cord injury. The signs are different.
What happens then?
Initially, especially after spinal cord injury, there's a period of spinal shock. Below the level of the lesion, you see paralysis, but also, paradoxically, areflexia and reduced muscle tone, hypotonia. This can last days to months.
Then it changes.
Yes. Spinal shock gives way to spasticity. This involves increased muscle tone, hypertonia. Muscles feel stiff and exaggerated stretch reflexes, hyperreflexia, like a really brisk knee jerk. This happens because the spinal circuits below the injury become hyperexcitable without the normal modulation from the brain.
So loss of reflexes versus exaggerated reflexes can point to different locations of damage.
Exactly. It's a key part of the neurological exam.
Okay, we've covered basic circuits, reflexes, rhythms. Now, how does the brain organize information spatially? Using maps.
Yes, the brain uses maps extensively. Think of them as abstract representations of specific information, laid out spatially across a sheet of neurons, kind of like a geographic map highlights features.
Like the body surface being mapped onto the brain.
That's a classic example. Sensory receptor sheets often form direct spatial maps. Skin receptors create a map of the body surface, somatotopy. The retina creates a map of the visual scene, retinotopy.
But not all sensory maps represent space.
Correct. The cochlea in your ear maps sound frequency along its length, not where the sound came from. Olfactory receptors map chemical types. But a key principle is that even a single sensory surface, like the retina or the skin, gets mapped multiple times in the brain.
Multiple maps of the same thing. Why?
Each map might emphasize different features or aspects of the input. And the brain can even construct maps of things not explicitly mapped at the receptor level, like calculating sound location.
Let's take vision. How does that map work in the cortex?
Okay, visual info goes retina thalamus, the LGM part, primary visual cortex, area V1. V1 contains a detailed retinotopic map.
And it's laid out. How?
Two key things. First, it's contralateral and inverted. Your left visual field maps onto your right V1, right field to left V1, and the upper part of your visual field maps to the lower part of V1, and vice versa.
Due to the optic chiasm crossing.
Exactly. Second, there's a magnification factor. The map isn't evenly scaled. The fovea, your central high acuity vision area,
Has way more brain space.
hugely magnified. It takes up maybe half of V1's area, reflecting its density of photoreceptors and its behavioral importance. We use our fovea for detail mark.
And V1 is just the start, right? You said multiple visual areas.
Over 25 distinct visual areas in humans. Nearly half the neocortex is involved in vision. Different areas specialize in processing shape, color, motion, location, etc., all in parallel. But they're massively interconnected, integrating everything into our seamless perception.
Are there even maps within maps in V1?
Oh yes. In layer 4 of V1, input from the left eye and right eye terminate in alternating stripes, like zebra stripes if you look down on the cortex. These are ocular dominance columns.
So segregation by eye input. Anything else?
In layers 2 and 3, there are these distinct patches called blobs. Neurons in the blobs seem specialized for processing color information. They project to specific zones in the next visual area, V2.
And V2 has stripes too.
Yep. Thick and thin stripes in V2 receive different inputs, like blobs feeding the thin stripes, and process different features, like motion or form. It's incredibly intricate organization.
Okay, let's switch to touch. The somatosensory maps. Penfield's homunculus.
The famous little person. Penfield electrically stimulated the cortex of awake neurosurgery patients and asked what they felt. He mapped out the representation of the body surface on the primary somatic sensory cortex, the postcentral gyrus.
And it looks distorted, right?
Very. It's often drawn like a figure draped over the cortex with huge hands, lips, and tongue, but a tiny trunk and legs.
Not entirely continuous either.
Right. The hand area often separates the head and face areas. The key is that the scale reflects sensory receptor density and behavioral importance. Fingertips and tongue are highly sensitive and crucial for exploration and speech, so they get lots of cortical real estate.
And this varies between species.
Absolutely. Rodents have enormous cortical areas devoted to their facial whiskers, which they use constantly for navigation.
And like vision, there are multiple somatosensory maps.
Yes, the somatotopic order is maintained through several stages: spinal cord, brainstem, thalamus, and there are multiple distinct maps within the cortex itself.
What about motor maps? Are they similar?
The primary motor cortex, right next door in the precentral gyrus, has a modal map that's remarkably similar in layout and magnification to the sensory map, especially for the head and hands.
But it's not quite a simple point-to-point map for movement.
Not really for fine movements. While gross mapping works, stimulating a point for, say, finger movement actually activates a distributed network of neurons. It's more about representing movements or goals than just individual muscles.
And sometimes maps overlap. Sensory and motor.
Yes, a great example is the superior colliculus in the brainstem. It has aligned maps for vision, hearing, touch, and motor commands related to orienting head and eye movements. They all work together seamlessly.
So, maps are everywhere. Why? What's the advantage?
Good question. Several ideas. Maybe efficiency keeping functionally related neurons closed minimizes wiring length. Maybe it aids learning new bineurons firing together, strengthens connections via Hebbian plasticity.
Could it simplify development?
Possibly. Easier to set up ordered connections. It might also make certain computations easier, like using local inhibition for edge detection and vision.
But these maps aren't totally rigid, are they? You mentioned plasticity.
Critically important point. Maps are dynamic. First, the map is not the territory. A single point stimulus activates a group of neurons, not just one. This distributed coding provides robustness.
And they can change.
Dramatically. Maps reorganize based on experience, learning, and especially after injury. If you cut a nerve supplying, say, a monkey's hand, the cortical area that used to respond to that hand will, over time, start responding to input from adjacent areas, like the face.
Wow, the brain rewires itself.
It does. This plasticity is fundamental to learning new skills and recovering function after damage. Macs are constantly being updated.
Incredible. Okay, final topic. Temporal representations. How the brain handles time.
And it does so with incredible precision, especially for hearing. Resolving time differences down to microseconds is key for figuring out where a sound is coming from.
How do we localize sounds? Let's start with up versus down, elevation.
For elevation, you actually only need one ear. The folds and shape of your outer ear, the pinna, cause sound waves to take slightly different paths to your ear canal, one direct, others reflecting off the pinna.
Creates interference.
Exactly. These interference patterns create notches and peaks in the sound spectrum that vary depending on the sound's elevation. Your brain learns to interpret these spectral cues.
Clever. What about left versus right, the horizontal plane, or azimuth?
That needs two ears, and the strategy depends on the sound frequency. For high frequencies, say above 2 kilohertz, your head casts a sound shadow.
So the sound is louder in the ear, closer to the source.
Right. The brain measures this interaural intensity difference, IID. It's largest when the sound is directly to one side.
But low frequencies bend around the head.
They do. Long wavelengths diffract easily, so there's hardly any intensity difference for low frequencies below maybe 2 kilohertz. For those, the brain uses a different cue. Interaural time difference, ITD.
The arrival time difference.
Precisely. A sound from your right reaches your right ear slightly before your left ear. Even if the sound source is just slightly off-center, there's a tiny delay, maybe microseconds to a maximum of about 0.6 milliseconds if it's directly to the side. The brain measures this incredibly small ITD.
Though both cues can be ambiguous.
Yeah.
Like front versus back.
Yes, a sound straight ahead gives the same IID and ITD as one straight behind. Head movements help resolve that ambiguity.
Okay, measuring microsecond time differences? How on earth does the brain do that?
It's a really elegant mechanism worked out largely in the brainstem auditory pathways. It involves neural delay lines and coincidence detectors.
Sounds cool. Where does this happen?
Key structure is the medial superior olivary nucleus, the MSO, in the brainstem. It receives input from both cochlear nuclei, which get input from each ear.
So it can compare signals from both ears.
Exactly. And crucially, neurons in the cochlear nucleus fire action potentials that are phase-locked to the waveform of low-frequency sounds, preserving the precise timing information.
Okay, phase locking.
Then, neurons in the MSO act as coincidence detectors. They only fire strongly if they receive excitatory inputs from both ears at almost exactly the same time.
Simultaneous arrival. But the sound arrives at the ears at different times.
This is where the delay lines come in. This is Jeffress's model from 1948. Axons bringing signals from the cochlear nuclei to the MSO neurons vary systematically in length.
Acting like different lengths of wire.
Kind of. An axon from the ear that receives the sound first might travel a longer path to a specific MSO neuron, while the axon from the ear that gets the sound later travels a shorter path to that same MSO neuron.
So the delays cancel out.
Precisely. For a specific ITD, the different axonal travel times compensate, causing the signals to arrive simultaneously at one particular MSO neuron, making it fire.
And different MSO neurons have different combinations of delay lines.
Yes. Each MSO neuron is tuned to respond best to a specific ITD, and these neurons are arranged systematically across the MSO, forming a spatial map of interaural time difference, which corresponds to sound source azimuth.
Wow. A map of time, essentially.
A computed map of time difference, which represents auditory space. In mammals, it seems synaptic inhibition also plays a sophisticated role alongside, or perhaps instead of pure axonal delay lines, but the principle of comparing timing via coincidence detection holds.
So these sound maps are computed by the brain, not directly reflecting the sensory organ layout.
Exactly, unlike the retinotopic or somatotopic maps. And then higher up, like in the inferior colliculus, information about ITD and IID is combined to create an even more complete spatial map of the auditory world. It's another example of hierarchical and parallel processing.
Okay, that's a lot of ground covered. Let's try to pull it together.
We went from the basic elements, inputs, outputs, interneurons making up circuits,
To simple spinal reflexes, like the stretch and flexor reflexes, including how the brain controls them.
then to the idea of central pattern generators in the spinal cord creating rhythmic movements like walking.
And finally explored how the brain uses spatial maps, like for vision and touch, with their magnification and plasticity, and computed maps for things like sound localization using incredibly precise timing circuits.
It's all about how these interconnected neurons process information in space and time to generate behavior.
Remember, if you're studying this, physiology is about connecting the dots, seeing that big picture, how these systems interact, before getting lost in the weeds.
You've honestly taken a huge step just by working through these concepts. You're building that crucial foundation. Keep asking questions. Keep tracing the pathways.
You absolutely can master this material. Don't get discouraged by the complexity.
Think about it, this constant mapping, remapping, computing.
Yeah.
It allows not just survival, but learning, adapting, thinking. It's an incredible dynamic system.
Truly is. You're part of the Last Minute Lecture family. We're here to help you dive deep. Until next time, keep exploring, keep
And you
learning.
keep asking, what does this all mean?